There we go. Yeah. All right, cool. Yeah, so no, it's literally just a chat over coffee. I hope you bought something because I have nothing for you, although there is a machine over my shoulder,
All right.
which I used. Um, no, the whole point was, I just realized that most of the good science you have doing these off the cuff discussions. And during the pandemic, Blaise, Frederick, and I would just every Tuesday that sort of built into a conference which we ran for a couple of years, and we might do again next year actually. And it was just talking about things that were of interest to us, because we had time on our hands. And you suddenly realize that this is actually when 90% of the good stuff happens, like it's in the coffee breaks between the
Yeah.
sessions, right? You get
Yeah.
stimulated by what you hear, and then or you rip
Yeah.
a paper apart, and then you have the insights, right? So that was the
Right.
point. So I'm just hoping that sort of magic happens here. And here in this particular case, I think 99% of the people who would ever watch this have no clue what MPI stands for. Max
No,
Planck something or other,
that's
right?
yeah, they're
So it's a way to introduce people to different technology. There's obviously the human angle that Larry Wald is fronting, right? Which I have mixed feelings about because I think it's just a vascular issue. We'll talk about that. But then I think something that you and I spoke about offline a few weeks ago was the lymphatics, and I'm especially curious about that. I think there should be something on the animal side that would make sense for MPI, and it doesn't really make a lot of sense on MRI, I think. So that's it. So let's kick off at the beginning. Do you want to try and give a quick overview of how it works?
sure, sure. So magnetic particles. So first, I have like a 20 odd year history, 25 years history in MRI. And the good news is MPI is a lot simpler. Think back to before you had the chip on your credit card, you just had a little magnetic strip and you would slide it across that magnetic chip to read the bits on the chip. It's actually a lot like that. What we do is we use the same super paramagnetic magnetic particles that actually were introduced by Paul Lauterbur in 1986 to the MRI field. And they're intensely magnetic. They're so magnetic, but they're safe. And so we actually directly detect the magnetization from those particles. It's already used sometimes in MRI, where they're detected mostly via a dropout in the image, a so-called T2 star dropout. But instead of actually looking for a small dropout near the particles, we actually look for a bright signal directly from the particles. And it turns out to be so much more sensitive because it's about 22 million times stronger signal from the particle than what you would get from the NMR signal around the particle.
And it's positive contrast, not negative.
Yeah, so it's positive contrast. You don't have all the confounds you have with negative contrast methods, and that's actually very beneficial. Probably the most important thing with positive contrast in the fMRI setting is for CBV and CBF, right? Because in anytime you're trying to do a cerebral blood flow measurement, it doesn't matter if it's with X-ray, CT, or MRI, you're always essentially taking two CBV images separated in time and subtracting them to get some form of a discrete, flow map. And the problem with that is that we only have about 5% blood volume. And then so you isolate your contrast agent, whether it's gadolinium or it's iodine, to that blood volume. But then you have 95% of the signal in the background is moving around just due to respiratory and cardiac motion. And so that sloshing in the background is going to be different on those two acquisitions. You're going to get a substantial subtraction artifact that has nothing to do with your blood volume and everything to do with cardiac and respiratory motion. And so we don't have that because we don't get any signal at all in MPI from the background tissue. So all we see is the signal
Let
in
me ask
blood.
some of my technical questions, then we'll go back to the sort of background physics, just so people can. I think I have a pretty good cartoon of understanding myself, but it'd be good to test it. So clearly you're doing particle counting essentially, right? If
That's right.
ultimately,
Yeah,
and
that's
then so just like you would, like a Geiger counter and beta particles or something, right? but
That's
this
right.
is just with magnetism. So you're going to count the particles that are present. So CBV I get, right? So if it goes from 4% to 5% to 6%, you get a proportional gain in
Yeah.
the signal.
Yep.
But how do you measure cerebral blood flow?
So we would do it almost exactly the same thing, and we have done this already, where you just take two frames separated by a small difference in time, and then you can subtract them. And we can collect, we can take a full 3D scan in 30 seconds, and then we could subtract. We could do, we could interleave, take part of the 3D scan, just like we do an MRI, and then we could do a subtraction over a shorter time frame.
Is there any way you can image flow directly or indirectly as a sort of a time of
Right.
flight type method?
You could certainly do some time of flight type methods. Absolutely. We don't have, like in MRI with a gradient, you can induce phase or, even frequency changes. So we don't have that effect. Everything in MPI is phase synchronous. And so it's a huge advantage. We can image, right near air and water. There's no artifacts whatsoever in the lungs in MPI, which is, of course, a difficult part, near the sinuses. If they're filled with air, then you can actually get a non-specific T2 star dropout that has nothing to do with activity and everything to do with the fact that you're close to the air. So we don't have that problem, but we don't have like a direct encoding method currently for velocity.
Okay, okay, I'll have to give that some thought. So as I understand it, you have that, just to sort of give a quick background on the physics. As I understand it, you have a saturating field, which I think this is like a Z squared term, something like that, like the old TMR early sort of point resolved spectroscopy methods from back in the late 70s, early 80s. So
Right. The
right.
so-called sensitive point method.
Yeah, exactly. Sensitive point. So you destroy you saturate the magnetism for these particles everywhere except in a point. Now we can think of that as a voxel, which we can raster in space however we want, right? And then
That's
you've got to excite transitions of at this field free point. And this is where you have your second magnetic fields, which are the ones that are oscillating. And then it's not a tuned, it's not the electronics is tuned, but it's not a resonant phenomenon. So you choose the frequency you want to operate at, and then you excite spin flips in these particles in that field free point. And that's what you measure with your, and then after that, it gets complicated. You look at harmonics rather than the direct pass through, blah, blah, blah. It's engineering, wave
That's
hands.
right. That's right.
Hands. Right. Okay. Magic
Right.
happens. Right. It's what you buy. It's what you buy the social. Right. And okay. So given that, so you're exciting and detecting at the same time, which was an important point, important difference to MRI, where we separate the transmission and the
Right.
reception a little bit. But you're also not necessarily preparing the spins. But going back to the flow idea, would it not be possible to do some sort of preparative step to try to sort of semi-induce the nudge the transitions towards the spin flip so that you do get a meaningful temporal difference? Because this is so fast, it's at the speed of light.
Yeah.
So flow is very slow.
Yeah. So, potentially, we haven't worked on that. Nobody in the MPI field has worked on it. So it's a good challenge. I like that. It what normally happens, you can think of it a little bit like adiabatic pulses and MRI.
Right.
We never ever let go of the magnetization. And so you could imagine, sort of you know, uh, trying to prepare the magnetization in a way where it would flow would change it, but we don't have any hardware to do that currently.
So right now in the field free point, we just stick to the point because it's easier conceptually. You're keeping everything adiabatic. So when you're doing these 3 kilohertz frequency transitions, you're trying to make sure that they stay aligned along the effective field, right?
That's
So
right.
relaxation
Right.
doesn't happen essentially, right? You can
That's right.
ignore the
Yeah,
relaxation. Yeah.
that's right.
Okay. So very
And
similar to adiabatic theory.
it is very similar. And all SPIOs in the field of view are always adiabatic, always.
Okay.
And that's good in some ways, but as you said, it has the problem that we have direct feed through. And so one of the clever things that came from the, Gleick and Weisenegger, the originators of the idea, was to make sure that the direct feed through was only at the first harmonic of the excitation, typically 20 kilohertz. And so everybody tosses that 20 kilohertz out because it's so contaminated with direct feed through from transmit to receive. And that's even after we go through some pretty tedious direct feed through cancellation with gradiometer detection. But it's still going to be a million times bigger than the signal you get from the patient SPIOs. And so everybody tosses that out. And so the image reconstruction problem is to just reconstruct the image, given that you're missing some of the information, a very minor piece of it. And it's actually very straightforward, the image reconstruction. The math looks horrendous, but it's actually very, very simple and very, very fast.
Right. So on speed, I mean, various people quote various numbers. We should probably sort of specify what we're talking about. Let's say rodent. In your papers, you typically would claim 40 or 50 frames per second, something like that. So and a frame is essentially a 2D slice. Is that about right?
Yeah, you can do that speed. I mean, everything, of course, you always match it to the application. Yeah. So when we're doing, we have done full 3D in 30 seconds, for example. We've done that in the past. But if we're doing a normal study that doesn't have any cardiac or respiratory motion, then we're often doing something where we want more signal to noise ratio, and then we'll take 5 minutes for the whole scan.
So yeah, so right, and then I think that's important for people to take away. Right? These numbers are not dissimilar to fMRI or MRI numbers, right? 3D.
That's right.
Typical.
That's right.
And then resolution 1 to 2 millimeters is a reasonable number for you guys. And so if you're talking about covering something in multi sections of 2D, then typical, very EPI-like numbers, right? So typical sort of spatial-temporal characteristics. And I think in Larry Wald's recent paper last year, where they did the CBV imaging in the mouse and the rodent, might have been a rat, with hypercapnia, I think they were doing a frame every 5 seconds, and it might have just been a single slab, but in any case, and they were doing 10-minute periods of hypoxia, of hypercapnia. So again, very typical to what you would do with a rodent application.
Yeah,
in MRI.
That's right, and the big difference between MRI and MPI on the specifications is related to the tracers. So if you use a gadolinium tracer, manganese chloride, whatever tracer you would like, an MRI, maybe even 19F, it doesn't change your spatial resolution per se, right? It might change your signal to noise ratio, and then you can trade that off for resolution. But in MPI, the tracer itself, as well as the gradient strength, those two things determine your spatial resolution. And so if you can get a tracer that has sharper magnetic resolution, that translates to better spatial resolution. And that's not normal how we think about MRI. If we want to do, the spatial resolution of a blood vessel with gadolinium, and it is pretty much identical to the spatial resolution outside the blood vessel.
Gotcha. Okay. So that's that sigmoidal.
Exactly. But we would love a particle that was saturating instantaneously.
Of course. Right.
And so we're just now, we just now have started out, a UO1 grant with the NIH to develop a particle that experimentally we've demonstrated up to 30-fold resolution improvement over, commercial grade SPIOs. But it's not yet ready for in vivo use, and that's what we're doing, is bringing in some chemists and a big collaboration to get that to happen.
These SPIOs, those are marketed anyway, right? That's the is that the Reservist, or is that getting?
So it's related to Reservist, but it's actually marketed under the name VivoTrax.
Oh, okay. And what's a typical resolution using that today, or is it also the distribution of particles sizes in this stuff? because MRI.
There's always a distribution, but a good figure of merit is what you said about a millimeter
Oh
resolution.
wow. Okay. Yeah. Yeah. Okay. Which for if you can do that in humans,
If you could do that in humans,
you're
yeah,
banging on the door of anything that
right,
we
right,
you need a
right,
70 to beat that,
right.
right?
But keep in mind that that's 1 millimeter in a very strong magnetic field gradient. So, that's in a 6 Tesla per meter gradient.
Sure, but unless you're moving in that, it's not going to be a healthy.
Absolutely right. And outside every 3T MRI scanner, just outside the magnet, there is about that order of
Yeah.
magnitude gradient.
Yeah.
And so
Yeah.
humans do walk through it slowly, hopefully,
Yeah.
and it's safe. Um, so right now, people are building human MPI scanners. It's still very much, an R&D project to get one out and make sure it's FDA approved and all that. And so there's 3 companies that are working on this. At my own startup company, Magnetic Insight in Alameda. Bruker, of course, very famous for preclinical MRI. They're working on it. They're not. I don't know that they're working on the human scanner, but they're very active in small animal imaging. And there's a newer startup company in Germany called Pure Devices.
Okay. And did Philips hang this up? They
So
built one, didn't they?
Yeah, so Philips was, hands down the leader for the first, maybe 8, 10 years of the field. Gleick and Weizsäcker were there at the time. But I think Philips decided maybe 6 years ago, something like that, 5 years ago, they decided that, it was the R&D elements might, be too long. And so they put most of those engineers into the MRI field.
Gotcha. Okay. Yeah, I mean, I'm going to hopefully I'll bug Larry to coming on here and chatting about this stuff too, because I mean, I see the non neurovascular imaging applications. Clearly, once you get something that can tag, in lungs or in cancer,
Yeah.
various other, right? I think there's a lot of good medical applications. The moment you've got good tracers, you've got a good target.
Yep.
Exactly by that. But that's so almost orthogonal to what most of the neuroimaging community would be interested in. And that, I think, is probably what got Larry funded it in the first instance, through the Brain Initiative. But it looks to me like it's just another vascular imaging method with all of the same
Yeah.
issues of physiological
Right.
confounds. He's going to have autoregulatory issues. He's going to have non-stationary blood gases modulating CBV, right?
Yeah.
I don't see, and also he loses the venous side, so he gets most of the arterial side, which is great. He might get spatial specificity down to the vascular bed, but he loses everything downstream, which we get for free, more than for free in fMRI. So still not sure that it changes the equation much. Just makes
So,
an
yeah,
imaging
yeah.
experiment much harder.
So the big applications that we've pursued in my lab at Berkeley are, some of them are, cell tracking, like tracking CAR T cells, tracking and CAR NK cells to a tumor. You could find out in a few days whether the treatment is efficacious, whereas it's not uncommon that those treatments are, may not be totally effective, but the physician won't find out for months with a follow-up FDG PET. And so we can actually do a functional test of the CAR cell, of the CAR T or NK targeting, which is the whole reason that for those treatments. And this is extremely exciting.
How long
So,
does the tag, how long do your particles live in this case? Do they indefinitely?
so almost indefinitely. So we would, while you're doing the CAR manipulation outside the animal or the patient, ultimately, you're already doing the CAR manipulation. Then there's dozens, if not hundreds, of these now, and they're really trying to figure out how to target a solid tumor. Most of the success with immunotherapy has really been with bloodborne tumors like leukemia, and whereas solid tumors, there's a lot more barriers to getting the immune cells into the tumor. They have to go through the endothelial layer, of course, in circulation, and then they have to find where the targeted tumor is. So it's still one of the biggest challenges, and 90% of tumors are, in fact, solid tumors. And so, but hands down, the most exciting part of oncology right now, if not all of medicine. The other applications that I see, they're all, mimicking SPECT and scintigraphy, right? Which are two modalities that we don't think about as much in the MRI field, but SPECT and scintigraphy are amazing technologies. They have 100 nanomolar sensitivity, right? And MRI is really good at if we're 55 molar concentration, which we are, depending on how much coffee we had today. But 55 molar sensitivity is great. But if you want to do a tracer like we do in SPECT and scintigraphy, you need that sub-micromolar
Why
sensitivity.
is SPECT so better, much better than PET? Just because you've got two photons?
Um,
Two
no,
pulses?
PET is probably a little bit better and
Okay.
a little bit more sensitive. The 511 keV photons are more energetic than 140 keV photons,
Right.
and you do have the two photons. It's not a simple physics equation, right? Because all of it depends deeply on how many of the photons you collect.
Mm.
And in PET, we're not using collimation, and so in PET, you're actually detecting a lot more of the photons, and that's where a lot of the SNR comes from PET. But MPI can actually do a lot, not all, but a lot of scintigraphy and SPECT, but with zero radiation. And that's not even the most compelling piece of this. I would argue the most compelling reason is kind of subtle. If you let's say you had a flight to the UK and you got stuck in the air for 18 hours, and you're not moving your legs around, you could develop a deep vein thrombosis, and that could lead to a pulmonary embolism. That's an emergency diagnosis. One of the tried and true methods of diagnosing this is what's called a VQ scan, a ventilation perfusion scan. And it's done all the time still. Because it's kind of slow and clunky, it takes 3 hours. And that's partially because you have to do the radiochemistry to target the vasculature in the lungs. And you have to do that just in time. You can't label it on the weekends or in the factory, from the drug manufacturer, because the radioactive decay is always ongoing. So you have a PhD in chemistry who's in every nuclear medicine department, and they're wonderful humans, but it takes an hour before every study can be done. And we don't have that problem because we don't have any radioactive decay. We don't have any decay at all of the SPIO. We've actually imaged for 87 days nonstop after one injection. We've been able to track stem cells for 87 days in vivo. And so the factory will do the targeting. All of it will just come out of the fridge. You don't need to do that hot chemistry labeling. Just inject, you're ready to go. And so
it can be shipped across the country,
no
manufactured
problem.
in one place or another country.
Yeah, and it's just
stable, safe.
totally stable, safe. You don't have to put a magnetic field on it or anything. It's just good to go.
Right.
So it took me a while to appreciate that advantage. It is extremely practical, but all of a sudden, deep vein thrombosis and, well, pulmonary embolism, a gastrointestinal bleed, those are all done primarily with SPECT and scintigraphy. Those are emergencies, right? If you have a GI bleed, they want to intervene immediately. And so waiting 3 hours or at least 2 hours for a scintigraphy study is not a good idea. And so even more compelling than the lack of radiation is the fact that you could do the injection in 5 minutes. You can get your first study, no problem. And anybody can do it. So you could do it at night. You could do it on the weekend. Most of these nuclear medicine studies are not done on the night or weekend because of the sort of labor of hot
And I'm
chemistry.
guessing they also are done, not like with strokes. So you have, these 100 and something stroke centers. If you happen to live more than 4 hours away, which a lot of the population of the US does, good luck with that. By the time you get to the stroke center, the actual stroke care, you're probably already well past
That's
most
right.
people's
That's right.
treatment.
And
window.
so the big future application is, I think, of MPI is stroke. And that's the big neuro, application is stroke. And of course, the big challenge of stroke imaging is giving an ironclad proof to the clinicians that it's ischemic or it's hemorrhagic, because there's a great treatment for the 85% of strokes that are clearly ischemic. You can give TPA or another clot buster, and then the patient might go home all happy and normal, right? Because you get rid of that clot in the 3-hour window you have, or 4-hour window now, and everything is resolved, right? If it's a hemorrhagic stroke, the treatments are much more limited. We don't have a treatment like TPA that's really, really awesome. Fortunately, only 15% of strokes are hemorrhagic, but you cannot give the TPA to a patient that is having a hemorrhagic stroke, because you can kill them.
Yeah.
And so finding out in a very, very short time if it's hemorrhagic or ischemic is probably the biggest diagnostic imaging challenge in any hospital. And MPI could do it. MPI could do a very, very good job. We could do a really robust cerebral blood flow, and that could help. But there's a lot of different sort of more pulse sequence type methods we could do to try to tease out whether it's hemorrhagic or ischemic.
Yeah, I mean, I spent a lot of times, you and I years ago, we were talking about this for low field too. I spent a lot of time in this space, and I still do. You've got, if you look at the MRI, state of the art with things like diffusion perfusion mismatch, and flare and diffusion for defining the penumbra, and then looking at flare, and there's lots of with lots of deep learning too now. It seems like they do a pretty good job of classifying these cases pretty well, but yet it doesn't seem to move the needle in terms of patient care because of workflow. Nothing to do with the radiology per se, but to do with who owns the scanner and where the scanner is. So the ER doesn't have the scanner. It's an outpatient, or it's across the hospital. And so
Right.
they do what they always do, which is CT, maybe CT angio, and
Right.
then they make a decision based on clinical science, and you've got 15 minutes.
Right.
That's it. So
Right.
that's the part that always worries me about this, is even if you have a killer solution, technical solution, you've now got to insert this into the workflow, and it has to be super fast and not mess anything else up that they would do anyway.
No, you're absolutely right. And then it is going to take a long time. But there's a reason there's an X-ray, there's an ultrasound, and a CT scanner in the emergency room almost everywhere in Europe and the USA. They're bulletproof. A patient can literally have a bullet and still get a scan.
Right. Yeah. No contraindications. Crucial. Especially
Crucial.
if they're not ambulatory, if they're unconscious,
Right.
polytrauma.
Yeah.
Right. Yeah. Yeah. No relatives nearby to do medical history.
Right. So there are a lot of reasons that that's there, and also there are economic reasons, right? But then all of the rest of radiology is scheduled, and so there's a real trouble to make anything work on an emergency basis if it is also scheduled, right? And so it's manageable, but there has to be enough, maybe a factor of 2 better than what they have now, for people to put in all the money and effort to, open up a new diagnostic imaging capability that might be used twice a day instead of 20 times a day.
Yes. Right. So the ROC curve has to really
Yeah.
fight.
Yeah.
And maybe also, obviously the edge cases are where most of the resources probably end up getting spent.
Yeah.
So yes, eliminating false negatives and false positives would be super, super crucial.
Absolutely right. Yeah. But I agree with you that, the MRI is pretty far along.
And
You know,
yet we don't seem to have moved the needle much on, there's a few clinical centers that do it. UPenn springs to mind, probably MIT or MGH.
yep.
A small handful, still even today, a small handful of people who do this. And they're
That's
all big
right.
research hospitals.
And I think it's going to take more innovation on the operational side as well as on the technology side. right? Because operationally, somebody has to have a dynamic allocation of those scanners when they're not being used for an emergency.
Yep.
And it isn't unreasonable to say we need a 20-minute call in from the ambulance, right? That's reasonable to just say, okay, clear out the MRI or, the MPI or the MRI, and I'm sure that a company like Hyperfine that's trying to put out a very low field permanent magnet head scanner, I'm sure that this is an area they would love to get into where they reduce the cost dramatically to put it in that scanner or in the emergency room. And it'd be very interesting to see if the resolution they can provide is adequate, with that low field strength. And I know that they're also working on, deep learning, machine learning algorithms to try to mitigate that. And that, because there's nothing right now in the emergency room for an MRI, I think it's interesting to see what it would do. And, maybe the 0.55 T MRIs, which are becoming popular, maybe that's a good niche for them.
Where, yeah, the risks are relatively low. Even
Yeah.
if you do have a contraindication, if it's a life-threatening condition and you're in your 70s, you just deal with it. Most these days, implanted defibs and pulse, deep brain stuff. These are all that they measure the lead length, right? Everything is put in so that it's essentially MR conditional up to 3T. Right. So actually, and I think people they've done this at low field too to make sure that they don't accidentally trip over the lead lengths in DBS, etc. So yeah, there should be few major concerns, even if you don't have medical history. Even if there is an implant, you just go with it anyway, because otherwise the patient's likely to be dead.
I mean, structurally or, organizationally, radiology is also part of the hospital, just like the emergency room is part of the hospital. So someone above those two should be interested in a logistical way of sharing that emergency room
No, and I
MRI.
think that's happened, right? I mean, that's why rarely these days do I have to reject somebody from a research scan because of an implant. Most people now have anything in the last 10 years, they have these little cards and it says it's MR conditional to 3T. Rarely, unless it's a very old one, the idea that we used to have to reject anybody with a pacemaker, sure, 20 years ago. But nowadays they're built, because they know these are the people who are most likely going to need an MRI. So they have to be conditional.
Yeah.
So,
and a lot of that is simple electromagnetics, right? At high frequency, but still it's not that complicated. And so I'm glad it's been resolved. I think a lot of the companies that, Boston Scientific and Stryker and all those guys who built those, devices, it wasn't obvious how, understanding the problem and certifying and everything was going to be beneficial to them. But I think that that's changed. I think that they did realize they needed that to be competitive.
Yeah, we know it would be a competitive disadvantage. Even if it was cheaper,
exactly.
no one would adopt that. I think a doctor would look at the liability and say no chance.
Exactly.
So on the technical aspects, then, going to do sort of stroke imaging, would you do CBV based images? You just look for dark spots, basically, no lack of communication into a region, and then.
I think I think CBV and CBF would be useful. I really do. And
so a dynamic picture, so basically a serial scan. You inject the thing, and you could do almost sort of pseudo real time, like you would with gadolinium first pass.
Exactly right.
And look to see how fast it gets to different locations.
Right. Yeah, and one thing that we can offer with MPI that I don't know that maybe you could get with SPECT is that 5% parenchymal, it's really capillary level perfusion. It's hard to see in most imaging methods that capillary level signal, because capillaries are 6 microns ID, and so nothing can really resolve the capillaries for radiologic imaging, and so they just sort of disappear, if you will. MPI, because we don't see any background tissue, even though we can't resolve the capillaries, we can still see the blush of the parenchymal tissue, and we can easily distinguish that 5% signal from the background, which is, just.
Okay. Well, I was going to say that wouldn't matter if you're just because most strokes are in relatively large vessels, but if you have a stenosis and you have poor communication, now you can quantify that. Now you have
Yeah, and you could see instead of the number, exactly right.
Now you can, not just where is it, but how much is it? How much is getting there? And that could be changing in real time. That would be, oh, that's nice. Yeah, okay.
Yeah, so you could see a whole big black area that would look just like the noise in the background, and that whole black area is not perfused.
Yeah. Okay. Yeah, that's cool. That's cool. All right. Yeah, that makes sense. I mean, in a way, you can sort of get that with gadolinium too, but because it's going the other way, it's negative contrast. It's much harder to
That's right.
quantify.
Yeah.
Right.
And the background signal never goes to zero in MRI. So
except where you don't want it to, right? Like
yes,
temporal pole, temporal lobes, and
or the lung.
It's already near zero. Yeah. Okay. That makes sense. So one other technical thing which I was worrying about earlier on when I was reading some of your reviews, is there a motion sensitivity if the subject moves, if the field free point moves relative to the some sort of blurring?
It's quite a bit less than MRI, like orders of magnitude less sensitive than MRI. One of the reasons that we're not sensitive to the 10 ppm magnetic field changes in the lungs is we're using a field-free line or gradient, right? And if you change that by 10 parts per million, you can't even see it. There's no motion change artifact.
It's already a cliff face, right?
Yeah. And so if the patient moves, sure, then things are going to jostle, but it's more like what you would see if you were, using a Xerox machine.
Okay. Just you things. That makes
or
sense.
ultrasound. It's the same as ultrasound. You're just scanning, and so then you would see a gap, and then you would see it normal again on the other side of the motion. And so there's no coherent ghosting. There's none of the stuff that happens when you're doing a case-based acquisition, because there's no phase in MPI. Everything is always phase locked. So we don't get any of the horrendous ghosting artifacts.
Interesting. Well, I'll be super curious to hear what Larry has to say. I wondered then why they did a rat hypercapnia rather than just do a first pass extraction. They could have done an MCAO model, right?
Yeah.
On one side. Maybe they have. Maybe they've got those data coming down the pike. But that would be sweet.
Yeah.
It's easier to do.
Yeah. I mean, the big thing the big question in my mind about fMPI, functional MPI is, all the applications I mentioned, they're potentially deadly. GI bleed, pulmonary embolism, stroke, those are all potentially deadly. And so there is always a risk with injecting a contrast agent. Doesn't matter if it's iodine or gadolinium or 19F, doesn't matter. Or scintigraphy, any sort of technecium agent. But the risk and reward are commensurate, right? There's, potentially deadly illness that you always want to diagnose before you treat. And so that's why I'm leaning more towards stroke than maybe straight, neuroscience applications where there might not be a deadly condition that you're really trying to tease out, right? Whereas with stroke, then the risk of the SPIO injection is easily, tolerated. And so that's my only concern with this basic science.
Yeah, one area. So you got me thinking now on the edge cases again. So we've got. I'm going to try and get a couple of people on to talk about pharma fMRI and just resting state writ large, and just as in a sort of a thought experiment. What should we be measuring and why? What other things? Because there are so many confounds in fMRI, and we really need independent measures to understand many of them. But a lot of the stuff that people would like to study with fMRI that are not strokes, they're not emergency medicine, but they're extremely important. Things like Alzheimer's, right? Various other dementias, and what that looks like, where based on maybe your risk profile, maybe your genetic history, or just behavioral things, you might well be a good candidate to have a tracer injected, because there may be you may want to take a compound or not take a compound
Yeah.
and it's going to be a relatively poorly conditioned experiment. A lot of these things that are just recently approved, we don't know what the side effects are long term. We don't know what the benefits are long term. We haven't had them in the field long enough. So maybe it's things like Alzheimer's that are more degenerative conditions where this would be a valid thing to think about.
No, I like that idea. And I think MRI might be preferred there, right?
Not necessarily though, because the time scale gets you, right? So I'm thinking, things like the locus ceruleus, where you've got all those, is it dopaminergic neurons, from these two little tiny nuclei in the pons, they distribute everywhere. And that's the current sort of bandwagon that all the Alzheimer's people are on, because that seems to be maybe related to overall brain activity. But to try, it's hard enough to image these things at all, right? Let alone their projections, and then they're brain wide. And now, if you're going to start to do something that looks like an fMRI experiment, everything is shifting. Your baseline physiology is shifting. Your arousal is shifting. Your blood pressure is shifting. Your CO2 is shifting. And electronics is shifting, or you've got heating, etc. So your time frame, seconds to tens of seconds, lots of things are changing. Minutes to hours, forget about it. Even ASL is good over that level of time frame. But if you give an injection on time 1, day 1, right, and then I come and I want to look up uptake of a region of the brain, let's say, we might have a hypothesis where there's some cycling going on, and there's some projection in this part of the brain. I need to look at what the overall net cycling is in this part of the brain over days to weeks, because that might be the only measure that makes sense for something that's degenerative over years to decades. Right. So now I wouldn't think fMRI has anything to say at all. If I would, I wouldn't believe it.
All right. I was thinking back decades. I think it might have been Robert Weisskopf who did a clever, change in oxygen. And I think he did I forget what the experiment is, but I think it was an fMRI type experiment with a oxygen change. And, it was kind of interesting. I liked it. I mean, it was, pretty safe and
as a contrast agent.
Yeah.
Joe Fisher and other people have been doing that recently too, and even doing as an alternative to gadolinium extraction for perfusion measures. It seems to work. Right?
I like it a lot.
Yeah. But that's that gets at perfusion over seconds to minutes, but that might not be relevant for Alzheimer's.
I agree. That's quite possible. But stroke, it could be useful.
Yeah. Well, I'm just wondering though. Maybe the fact that you have this longevity, I guess the crux though then is how, if it's an intravascular contrast agent, how long does it stay circulating before it washes out? That would be your
right. So we're assuming that with the CAR T and CAR NK that they leave the vascular system soon.
Yeah.
No doubt a lot of them are going to get stuck in the liver and spleen through normal, RES release. But the ones that we care about, the ones that actually are leaving through diapedesis and actually hunting for tumors, those are the ones we care about. And that, we could track them for months, no problem.
And they don't get, so it stays intravascular. It's not getting excreted by the
kidney. No. They would only stay intravascular for, well, it depends on the nomenclature to some degree. So we have done this experiment, and they stay in the capillaries of the lungs for maybe 2 days, right? And the reason is that the capillaries are 6 micron in the lungs. And so if you do an intravenous injection, a cell that's 15 microns is definitely going to get stuck in the capillaries of the lungs for a full day and a half. Then we presume that they're removed by the RES system, macrophages and neutrophils, which then migrate to the liver and spleen to try to excrete them. And so all the people working on stem cells have to deal with this delivery challenge. And same thing is true with CAR T and CAR NK cells. They have to somehow bypass the normal immune system if they want to target, and so they're smaller. Obviously, we would be taking autologous white blood cells, and they should be able to squish through, and they should be able to actually circulate for, maybe even days or more. And
And you can't. So could you tag red blood cells? Could you take somebody's
Yeah,
tag
there's
them into
actually
the
a
lab?
startup company that's doing that. And so, technetium 99 RBC, or RBC scintigraphy, that's what they do for gastrointestinal bleeding. So they take the patient's own red blood cells out, they isolate the red blood cells, they label them with technetium 99M, re-inject them, and then they do a digital subtraction angio. So they'll do one scan and then they'll subtract that from another one. And so if circulation's identical, they subtract perfectly because there's no phase artifacts in scintigraphy. And then if there is mismatch, it's due to some extravasation. So you've got to bleed. And scintigraphy might have worse than 5 mm resolution. It's nothing like MRI or CT angio. But it can detect the smallest bleed because of that incredible, 100 nanomolar, whatever, sensitivity that SPECT and scintigraphy have. And we were able to actually surpass that. We did some experiments on this, back in 2017, and we actually published, findings that MPI was already competitive with nuclear medicine, which is astonishing given that they're radioactive and we're just magnetic.
Mm-hmm.
So
That's cool. That's
so I like those kind of applications, but you're right that you do have to extravasate when you're targeting like a solid tumor or any of those conditions.
interesting. Yeah, nice. I mean, there's another cottage industry looking at blood-brain barrier leakiness with gadolinium compounds, and
That's right.
once it's
That's
in, it
right.
has to get out.
Yeah.
If there's nothing worse. It's not worse than that. I mean, you could imagine some sort of whatever these particles are encapsulated in, that could be uh, made attractive to a macrophage, right? So the macrophage comes
Oh
along,
yeah. No,
oh, yummy, yummy, thank you very much. And it's just hemosiderin underneath. Okay, thank you very much.
so believe it or not, 4 years ago, we published exactly that. We used an anti-LY6G antibody on a nanoparticle, and it happened to have a good MPI signature. And then we injected that into the tail vein, and it actually attached itself to neutrophils and macrophages in circulation.
Oh wow.
And so then we could actually see those, do like a functional, almost like an indium 111 neutrophil study, or white blood cell study, where we saw uptake into the bone marrow. We saw some uptake towards or activity much preferred towards like we used an injection to provoke inflammation, and sure enough we saw the white blood cells respond to it. And but it is very new stuff, and because we're stuck building scanners, we're also trying to build new tracers for MPI, and we're doing applications. We're kind of torn in
Right.
3 or 4 different directions.
Yeah, of course, absolutely. Yeah, you mentioned the chemists. Well, that's super fascinating. So yeah, you could imagine if somebody could get a good handle on something that would end up being rendered non-toxic if it gets into the brain. If there
Yeah,
is
yeah, yeah.
leakiness, then that's half the battle right there, right?
Oh
Because
yeah, 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 yeah.
it's the opposite of MRI, right? Because MRI, we're looking for stability so that it can be cleared by macrophages intact without it taking the chelate off and the gadolinium getting exposed.
Right, right.
You would do the opposite logic here and have it just chopped up and spat out, used up.
Yeah. And the new macrocyclic gadolinium agents are pretty darn good, and like
Yeah.
they're very, very stable now. And so the big problem 15 years ago with NSF seems to have largely been dealt with.
Yeah.
And so
They are
that's
still they are still negative contrast though.
yes, absolutely. Yeah.
Still
And
trying to find a signal void amongst signal voids in the MRI.
yes, yeah, no, it's not an
Really a
easy.
dynamic signal void.
Yeah.
I wanted also to ask you, we mentioned, a few weeks ago we were talking about lymphatics. Why has nobody yet tried the glymphatic experiment in an animal with an MPI?
Right. I think it's just that we haven't had people who are, sort of card-carrying neuroscience types enter into MPI yet. I mean, Larry Wald certainly very interested. He's the first to do that. But Larry is also, he's got a physics PhD from UC Berkeley, and so he, has been building his own scanner, right? And so,
He's interested in the technology.
yeah, and I'm the same way. I'm primarily an electrical engineer. I don't know what I am anymore, but um,
Almost retired, you were.
almost retired. You never know when you should have retired. And so I get excited about technology. I get excited about building scanners and systems and all that. And the applications are also very, very compelling, but I wouldn't say that's my A game. That's my A minus game. And from the big picture point of view, I think the work we're doing on developing high resolution MPI tracers, that's probably the single most important thing my lab is doing right now. And so that's why I brought in chemists like Greg Tikhonmurov and Phil Messersmith, and of course, Ben Fellows from our startup company, who's formerly a postdoc in my lab. Those are the really, the big things, because that would benefit all of these applications. Anything that could give us an order of magnitude improvement in resolution would really make it enabling to do a human MPI.
I see. Yes, I don't know that this would necessarily translate into a human. Maybe that's one of the issues. But then again, there are plenty of people doing these cisterna magna injections with Reservist, and then looking to see where this stuff goes.
Yeah, and I think that would be it would work. It would definitely work. And
so maybe it's just that the people who have the scanners don't have the neuroscience background, as you say. We just got to find. Hopefully, someone listening to this, they will talk to each other, because it seems like it would be trivial almost.
I'm happy to collaborate.
Okay, all right, good. I'll make sure I put a big old glaring sign on where Mike and Nedergaard or whoever is doing this stuff, they need to come talk to you guys.
Yeah,
because that would be super, super cool. I mean, yeah, I mean, that would be fascinating.
Oh yeah,
because a lot of this stuff gets very confusing, and it's hard to, again, it's the timescales, the MRI timescales. Okay, yeah, even if the particle exists in the CSF and it gets into the brain for a long time, you're still using it to knock out a short-lived seconds signal that you produce with the MRI. That is very difficult. Whereas if you just simply have the particles there and you can image them day after day after day after day, much, much nicer curve.
Yeah.
All right, cool. Then I might start to believe some of this glymphatic stuff. It's interesting literature to follow, but I don't know. Nobody seems to have got a good handle on it. So what else? Anything else before we? What else are you thinking of doing with this stuff?
No, I think that's the main thing, and honestly, MPI doesn't line up very well as a competitor to MRI, right? Because I mean, six orders of magnitude different concentrations, right? We're aiming, we can detect 1 micromolar concentration of SPIOs. MRI is like fabulous soft tissue contrast and resolution at 55 molar concentration of water. And there's just apples and oranges in terms of the strengths. Like MPI can't even see tissue. MRI is fantastic at seeing gray matter, white matter, CSF, unparalleled. Like CT can't even come close. So for the 90% of clinical MRI, MPI is just orthogonal, and we don't compete. We really do line up in MPI to competing with SPECT and scintigraphy. When we figure out how to target nanoparticles to tumors effectively, then I would say that MPI can start to compete with FDG PET. But currently, we don't have a targeting antibody or white blood cell, I think, that can beat FDG PET. I would be ecstatic if we could beat PET, FDG PET, but it's really, really good.
But isn't FDG PET looking just at metabolism through?
Mhm.
So it's
Yeah,
still looking at essentially just the perfusion levels. So
that's correct.
essentially, its time frame will drop away. For these
Yeah.
solid-like tumors, then you're still going to lose sensitivity there, just because
And
the
you're correct that a slow, growing tumor, often like prostate tumors are slow growing. And so FDG PET isn't as sensitive on those because that you're right, it's just measuring metabolic activity. And if it's comparable to the background, metabolic activity is hard to see it.
mm,
Uh, but there's enormous potential here if we could use white blood cells like CAR T, CAR NK, but instead of thinking about them as merely, or I don't want to say merely, as a therapeutic, obviously that's enormous and worthy of the Nobel Prize and immunotherapy. Um, um, Probably what I would love to do more than anything on the application side is to go back to this notion of theranostics, where, let's say we, continue with this anti-LY6G or another antibody, a nanoparticle, and we find out it targets neutrophils and macrophages. Those might be able to find a tumor where our own invented antibodies have failed, right? Because those white blood cells are so much bigger than the nanoparticle, right? And they can get in and out of blood vessels, even through the blood-brain barrier,
mm,
through diapedesis, even though they're orders, 15 microns, right? Whereas the nanoparticle could be 80 nanometers, and it can't get through the blood-brain barrier. But diapedesis, sort of snakes in between the tight junctions, and it can actually extravasate. And so I want to hijack the white blood cells to do the hunting for, cancer, maybe infection, maybe inflammation. And that would be replacing the indium 111 white blood cell study. Um, this, to me, has got a lot of good qualities because if you think about it, we actually inject. So when we do an indium 111 white blood cell study, it can take 2 days because you take out the white blood cells, you isolate them, right? Mostly it's neutrophils and macrophages in humans. Then you put in the indium 111 and you label them. The problem with it is the entire radiation dose to a so-called 70 kilogram patient is first being imparted to a 70 milligrams of white blood cells. That means the radiation dose to those white blood cells is literally 1 million times higher than it is to the patient. And I'm not feeling bad for the white blood cells, but we are relying on them being cogent.
yes, right. God knows what's happening to those things.
Right. We
All kinds of
punch it as
electrons
hard
getting
as
knocked
we
off.
possibly
Yeah,
can
yeah,
with radiation and then say, go ahead, go find
yeah,
infection.
yeah, yeah. Hopefully, you're still normal. Hopefully,
Yes. Right. And so that's neutrophils and macrophages, but CAR T and CAR NK or any T cell and any NK cell are actually more radio sensitive. And so I think the radiocytotoxicity is going to be one of the big things that comes out in MPI, that we don't have any of that, cytotoxicity. And so tracking white blood cells, even red blood cells, if we want, those problems are completely mitigated.
mm,
And then I think we might actually be able to greatly improve on, white blood cell in indium 111. And they don't like indium 111 as a tracer. It's got a 2.8-day half-life, but that's only because it takes very long for the white blood cells to meander through the body and find infection.
I think that, yeah, to me, that's the key of all of this is that it's the time constant, right?
Yeah. Yeah.
NMR is milliseconds to seconds.
Mm-hmm.
And at 70, T1's, it's gone, right? And then so your labeling is, we have this with perfusion imaging. You can label in the neck and you can get to the brain, but getting through to through the blood-brain barrier into brain water is already down 2 orders of magnitude. And good luck with that. even with radiation, you've still got these relatively fast half-lives. If you can get something that's days, weeks, months, and it doesn't care and it's essentially stable, that really does change the equation. There's no toxicity while it's going where you want it to go before you measure it. You're not required to do anything until it gets where you think it should be, and then you can measure it ad infinitum, which I think is just wonderful. I think it's really, really, really cool.
Yeah, I agree. I agree.
So, all right, well, cool. Thanks, man. I appreciate this. So let me
Good fun.
let me do the stop record thing if I can find it.